Good evening, everyone, and good morning, Professor Ramirez from Brazil. I'm Chanaka, a third year PhD student from Estonian University of Life Sciences. So today our guest speaker is Professor Marcel Ramirez from Institute of Carlos Chagas, Brazil. And Marcel Ramirez is a full professor from Carlos Chagas Institute, Brazil, and a fellow of a Brazilian Research Council. He holds a degree in biomedicine from the University of Antofagasta, Chile, a master degree in microbiology and immunology, and a PhD in biological sciences from the Federal University of Sao Paulo. He has conducted postdoctoral studies in the Federal University of Sao Paulo, London Metropolitan University, Royal Holloway College, Max Planck Institute, and German Trias and Pujol Institute. He has experience in parasitology with an emphasis on cell biology and immunology, working in parasite-host cell interaction. His laboratory focuses on characterizing extracellular vesicles derived from Trypanosoma cruzi and Giardia intestinalis. Today, his lecture will be on the topic of role of extracellular vesicles during parasite-host cell interaction. So, Professor Ramirez, the floor is yours. You can start.
Okay, thank you very much for your nice presentation. It's a pleasure to be here. I would like to be there next time. And it's an interesting possibility to have video conference through the world. So as you mentioned, my name is Marcel Ramirez. I'm a researcher from Carlos Chagas Institute-Fiocruz, and the title of my talk is Extracellular Vesicles during Parasite-Host Cell Interaction. We have a group, Extracellular Vesicle and Host Parasite Interaction Research Group in my institution, and I would like to talk about my talk. So first of all, I would like to say who we are, and later how we came to study extracellular vesicles in Trypanosoma cruzi and later in Giardia intestinalis. I will focus in Giardia intestinalis research in this talk. And so this is the historical building that was created, the Fiocruz, Fundación Osvaldo Cruz in Rio de Janeiro, for Osvaldo Cruz, 124 years ago. Now this castle is only for administrator staff, and we have 16 institutes distributed in all Brazil in 10 states. We have an institute in Africa, in Mozambique too. So our institution has hundreds of researchers and thousands of students, PhD and master degree students. And we are focusing teaching, research, innovation, producing vaccine, medicines. We belong to the Minister of Health of Brazil. And just to introduce my group, we work at the Fiocruz Institute in Paraná at the last 2.5 years. So this is my group. And Abel Sana is a student, PhD student from Guinea-Bissau, Africa, and is working with Trypanosoma cruzi. Isadora Rossi is postdoc in my lab. He made the master degree and PhD study with Trypanosoma cruzi EBs. Bruna Sabaki is a PhD student working with Giardia intestinalis EBs. Beatriz is an ungraduated student working with Giardia intestinalis. Leticia Bonato is a master degree student working with Giardia intestinalis. Sara Fusio is a master degree working with Trypanosoma cruzi and Giardia intestinalis. So we have half of the group working with every model. It's okay now? So all my life, I have been working with host-parasite interaction, trying to understand the relationship between host, parasite, and environments. And our focus has been neglected tropical disease, that we have a social and economical problem in South America, Africa, and Asia, and different points of the world. And I have been working with Chagas disease for a long time. That is a disease endemic in Latin America, and in recent years has been propagated for migratory movements to Northern America, Europe, Australia, and Japan. So the impact of the Chagas disease is we have 6 million people estimated to have Chagas in the world, and over 75 million people at risk, 30,000 new cases a year, and with 12 deaths per year. But the problem is that less than 10% of these people has been diagnosed, and is endemic and is propagating through the world. So the Chagas disease is caused for Trypanosoma cruzi, that is a flagellated, that is transmitted for insect from the Reduvidae family. And we have two kinds of stage of the disease. The acute stage is only for one person of the people infected. This is a very quick stage, and after 20 or 30 years, these infected people can be developed chronic stage with megaesophageal, megacolon cardiomyopathy, producing a problem and kill people. So the difficulty of the disease at the undetermined stage, where 50% of the patients are serology positive and are asymptomatic. So we have no good therapy, the toxic drug, and no vaccination. So every acknowledgement about the basic science in Chagas disease is important to think in new control therapies. So, but the model is very complex. The model is very complex because the life cycle is very complex, including invertebrate and vertebrate host, and 4 stages of the parasite. And the cycle started with the insect contaminated suck blood from the human patients through the feces, eject tripomastigol metacycle that ingress to the hot cells, invade cells producing the infection. And this parasite transform in amastigol form, that is the replicated form, and after transform in tissue culture derived cells that release from the cells and disseminate the disease. So many years ago, we decided to start with my group with the same question: how metacycline tripamastigol become resistant to lysis and able to produce infection in eukaryotic cell in this first time that the parasite is an intracellular obligatory parasite, need to resist to the innate immunity to produce the infection. And we started to analyze a mechanism of activation and resistance to complement mediated lysis. And for many years, we described a different evasion mechanisms, the expression of receptor for complement factor, the presence of inhibitor of serine protease, and the first evasion mechanism is invading to hot cells before being lysed by complements. So in this manner, the parasite could produce the infection. But in this time, 12 years ago, we asked about if the parasite could be produced at the first contact, the stimulation of release of extracellular vesicles at the first contact with the human, at the blood, at the contact with the blood. And so as you know, so extracellular vesicles are a membrane component released for most of the eukaryotic and prokaryotic cells. And for biogenesis, we can divide in two big groups. So the small EVs like exosome that are produced for endogenic endocytic pathway with multivesicular bodies releasing a component from 50 to 150 nanometers, and the microvesicles originated for plasmatic membrane that could be from 100 nanometers to 1 micra. And this is a cartoon to show the difference at the biogenesis at the microvesicles and exosome. So the exosome are producing permanently for multicellular multivesicular bodies, releasing a component from 50 to 150 nanometers. And the microvesicles are producing when the cell receive a stimulus that could be the presence of pathogens near to the plasmatic membrane or some injury of the membrane. And this stimulates the mobilization of intracellular calcium, activating calpain that cleave actin, and cleaving actin inhibit the flipase and flopase enzyme that are putting phosphatidylserine into the cells and stimulate the activate scramblase that put phosphatidylserine out the cells. And this activation produced the release of heterogeneous membrane component called microvesicle that has a size from 100 nanometers to 1 micra. So we decided to study what's happened in the first contact with the parasite, with the blood, and we designed an in vitro model to study the extracellular vesicle release in Trypanosoma cruzi. And for that, we used THP1 cell line that is a monocytic cells, and these monocytic cells represent the blood. And we used metacyclic trypanostic and tissue culture derived cell to have a contact in vitro. So we designed a methodology putting a relation from 5 or 10 parasite to 1 cell THP1 with 1 millimolar of calcium. That is a activator of microvesicle at 37 degrees and 5% CO2 at the incubator for 1 hour. And after this time, we centrifugate the parasite are removed, and we take the supernant, and then supernant is submitted to differential centrifugation to eliminate debris and to obtain the microvesicle by ultracentrifugation. In this time, we obtain the pellet and analyze the pellet by nanotracking analysis, by flow cytometry, proteomic analysis, different functional assays. And I will summarize years and years of work in Trypanosoma cruzi in just 2 slides. So we discovered that the contact between the parasite and the cells produce a release of extracellular vesicle, and these extracellular vesicles are able to increase the infectivity to hot cells are able to inhibit complement lysis by C3 convertase decay block. And this process was strain dependence in metacyclic triple mastigoid form. Other group has been contribution in Chagas disease with EBs, so showing that EBs carry different proteins that are parasite virulence. EB carry different RNA contents, and EBs capture for the cells and producing a remodelation of the gene expression of the cells. EBs modulate the host immune response, and the EBs are able to increase parasitemia and death the infected animals. So we continue with our question, trying to understand what's happened with the dissemination of the disease with the tissue culture parasite, looking for a chronic phase. So our question was, what is the role of the EBs in the chronicity of the infection? We simulate a model trying to understand what is the tropism of the parasite during Chagas, chronic Chagas disease. So for this, we analyze Viru, CACO2 cells, and C2, C12 cells that represent fibroblasts, intestinal epithelium, and myoblasts. And this, with this infection with the different strengths of the parasite, and looking for the production of BBs and the ability to replicate and to the excite of parasite from the infected cells to understand tropism and the modulation of the EBs. Basically, we have this model, so simulate the myoblasts with C2, C12, and simulate the intestinal epithelium with CACO2 cells. And we did a different infection and study the dynamic of the infection, the replication, the liberation of the parasite from the cells. And we collected the EBs during this process, understanding what is the effects of these EBs and the infection, replication, the liberation, and we analyzed proteomics and immune system activation. Just to show our preliminary results that we have included in a manuscript 2 months ago. So this is a proteomic study with an infection from myoblasts and intestinal epithelium with different strain for 2 hours and 24 hours. And we collect the EBs and we analyze by max spec. And we have a very interesting result looking that at the first 2 hours, we have a strong participation of the hot cells to share the EBs at the release of these EBs. We have more component from host protein than the parasite, and this effect equalized at 24 hours. If you compare the 2 hours and 24 hours at the different combination of the infection, we always obtain a higher concentration of protein at 2 hours than 24 hours. And if you look for the difference at the localization of the protein, most of the protein in the 2 condition are from cytoplasm, mostly 50%, and the rest are protein with signalization from nucleus, extracellular, mitochondria, and membrane, cell membrane. So we have no big difference between the localization. Summarizing this part of the trypanosoma cruzi, so we can see that proteomic results show that large EBs has different component depend from the myoblast or epithelial cell these EBs has been enrichment in different components. And the data show that we have an intense reaction from the 2 first hours. And this has a difference between the different cell lines, and results of parasite invasion and excitoparasite from cell show tropins and could be modulated for extracellular vesicles. And we are working at the lab at the moment, analyzing extracellular matrix component and tropies with different cell lines. So, but the second part of my work is talking about Giardia intestinale, trying to analyze the communication between the parasite and host cells during the interaction. And just to a brief summary about Giardia intestinale, is a protozoan flagellated, so it is a neglected disease, a diarrhea that is produced in underdeveloped country, and 280 million of cases annually, and is distributed in all the world. Giardia intestinale has 2 states, very interesting, trophozoite and cysts. The cysts are the infected form. The trophozoite has 4 nucleus, a different pairs of flagellum, a ventral disc that is important to attach the parasite to the intestinal cells. And this is considered the primitive, the first eukaryotic cell, because it has no mitochondria characteristic from other eukaryotics or mitosome, and has no endoplasmatic reticulum normal, and has no normal Golgi complex. So it's a primitive parasite. The life cycle starts with the cyst, that is the infected form, contaminate water or food, and for oral contamination, go to the stomach, and the stomach for the different pH release the cyst, release trophozoite, and this trophozoite adhere to intestinal cells at the small intestine. And this produce symptom and chronic disease, producing diarrhea. And for some signal, the parasite go to the form cystation again, and is eliminated by feces, completing the cycle. And so to produce diarrhea, you have an increased intestinal motility, problem with a small intestinal absorption of the nutrients, disruption of the intestinal epithelium. We have a reduced intestinal enzyme, atrophy of bilious, activation of host lymphocytes, and produce apoptosis in enterocytes. So we trying to understand what's happened during the first contact between the parasite with the intestinal cells, looking if these communication from extracellular vesicles are influenced at the first contact. Is these EBs are releasing to control the host immune response, and we have a transference of genetic information. For that, we establish a experimental strategy. At the first, we're looking for the liberation of EBs in vitro, and these EBs from the Giardia intestinale put in contact with intestinal cell like CACO2 cells, and we isolate the parasite again for isolate the EBs by differential centrifugation, and with the pellet we analyze for nanotracking analysis, protein dosage, functional assays. And we were the first group at the world to describe the release of Giardia intestinale EBs, And in this paper 7 years ago, we showed that the EVs increase the adhesion of trophozoite to caco cells, and these EVs are important, the lipid raft to produce the EVs during the biogenesis in Giardia intestinalis, and these EVs activate dendritic cells. And we continue with our research in Giardia intestinalis, looking for subpopulation, and we divided the EVs in exosome and microvesicles. And we used one inhibitor that produces an inhibition only the microvesicle, that is a peptidyl arginine deaminase inhibitor, that is an inhibitor that pass from the arginine to citrulline, that is very important at the metabolic from the parasite. And we got a parasite without adhesion when we treat with this inhibitor. So the parasite didn't adhere to the intestinal cells, and we used the microvesicle to restore the capacity of the parasite to adhere to the intestinal cells. Exosome didn't have any effect at the restoration of the parasite adhesion. So we analyzed the proteomics, and we have the different component in the small and large EVs, and for the first time, we have a different function from different subpopulation in protozoa. And we decided to understand what's happening in these first contacts, and trying to understand the communication between the parasite and the host cells. And for this reason, you know that the EVs could be internalized for the host cell for different mechanisms, and endocytosis mechanisms, maybe macropanocytosis, phagocytosis, just fusion with the membrane, plasmatic membrane, or activation for some factor at the plasmatic membrane. And so our interest is understand the balance between the biogenesis of the EVs versus the uptake. Uptake is the process that the EVs are internalized by host cells, and these could be different mechanisms: Clat3 mediated endocytosis, lipid raft endocytosis, macropanocytosis, caveolin mediated endocytosis, phagocytosis. So we decided to understand these mechanisms and this is a schematic about a schema about the different endocytes process. So we can divide in two big group: the dynamin dependent and dynamin independent. Dynamin is a JTPase, very important for the vacuole formation during the endocytosis. And you can divide the process in dynamin dependent, that are Clat3, caveolin, macropanocytosis. Caveolin, Clat3. These are the first mechanism, dynamin dependent, and dynamin independent, that is macropanocytosis and phagocytosis. And so we decided to do the same strategy, but now we have included 3 kinds of vesicles. So the second part of my talk is talking about Giardia intestinalis and extracellular vesicles. We're trying to understand the communication between this parasite and host intestinal cells. So Giardia intestinalis is a flagellated, belong to neglected disease, producing diarrhea. This disease is happened in underdeveloped countries. We have 280 million cases annually at the world. The parasite has two stage, the trophozoite that has four nucleus, a ventral disc that is very important for attach to intestinal cells. This parasite is has primitive organelles. The parasite has no mitochondria, has mitosome with no ATP production. The endoplasmatic reticulum and the Golgi complex are very primitive. Um, the other stage is the infected form cis that has a strong wall that is resistant to environmental condition. So the life cycle of the parasite start with contaminated water or food and disease entry into the by oral contamination to go to the stomach, and the stomach for the different pH release disease in trophozoite that go to a small intestine to attach the intestinal cells. And after some time, this parasite differentiate again in cysts, going to the colon and is ejected by feces to continue with the cycle. And this uh, presence of the parasite at the intestinal cell produce symptom and chronicity, and this disease is treated by imidazole derivative, and the disease start with the alteration and the intestinal motility, a problem with absorption at the small intestinal level. This produces a disruption of the intestinal epithelium, reducing intestinal enzyme, atrophy the villus, activation of host lymphocyte, and finally producing apoptosis of enterocyte. And this is the pathologic pathway of the diarrhea. We started to ask about what happened with extracellular vesicle during the parasite in contact with hot cells, looking for the communication between both cells and if there is some modulation of the host immune response or transfer of genetic information with the extracellular vesicles. And we have designed at the first uh, experimental strategy, that we analyze the production, the release of EVs from Giardia intestinalis in vitro, label the EVs, and after we analyze by functional assays. So we have the parasite in vitro for 37 degree, 1 millimolar of calcium. After this incubation period from 1 hour or 2 hours, we centrifugate, separate the cells, and the supernatant was submitted to differential centrifugation to eliminate debris and to produce microvesicles and exosome. And this pellet has been characterized by nanotracking analysis, protein dosage, functional assays, and proteomics. And we were the first group to describe release of microvesicles in Giardia intestinalis. And in this paper, published 7 years ago, we have shown that EVs increase the trophozoite adhesion to CACO2 cells, that these EVs to be produced are involved with lipid raft, and these EVs activate dendritic cells. We continue with our work looking for subpopulation of EVs that could be important to understand the function. So we have separated exosome and microvesicle, and using inhibitor of peptidyl arginine deaminase that is very important for the transformation of arginine to citrulline that is important in a metabolic pathway in Giardia intestinalis, we have got an inhibition of the release of only extracellular vesicles, large EVs, microvesicle, and not small EVs. And when we treated the parasite with this inhibitor, the parasite didn't adhere to hot cells. We have shown that when we added large EVs purified before in contact with the parasite treated with the inhibitor, the parasite are able to adhere to intestinal cells. And the only effect is from the large EVs and not small EVs. We have done proteomic analysis from a small and large EVs, looking that they have some market difference and other in common. And we trying to continue with our question, looking for what happened in this communication between the parasite and the hot cells. This is called cell communication that could be mediated by extracellular vesicles. And the vesicle release during this contact could internalize into the hot cell by several mechanisms. And this is our question. So to understand the balance about biogenesis of EVs from the different cells and the uptake from the eukaryotic hot cells. The possibility to entry into eukaryotic cell could be different kind of endocytosis. So clathrin mediated endocytosis, lipid raft endocytosis, macropinocytosis, caveolin-mediated endocytosis, and fusion from membrane from the vesicles. This is a scheme that shows and classify the different kinds of endocytosis, and we call associate the dynamic dependent or dynamic independent of this different pathway. Dynamic is a GTPase that is very important to the formation of the vacuole and the early endocytosis. So we have as dynamic dependent process clathrin and caveolin, and as dynamic independent process macropinocytosis and phagocytosis. So we started with the experimental strategy. By now, we were worried with three kinds of extracellular vesicles. Giardia intestinalis extracellular vesicle released for the Giardia properly, hot cell CACO2 host EBs released for the eukaryotic CACO2 intestinal cells, and the interaction vesicle produced during the contact of the parasite with the host cell CACO2 intestinal cells. So these three kinds of EVs can be stained for different lipophilic fluorescent to understand the uptake of the uptake process. So we have in contact during the incubation for production of the EVs, and we purify again by differential centrifugation, pelleting the cells at the first, and after the supernan is submitted to differential centrifugation to eliminate debris and to separate microvesicles at the first, exosome by ultracentrifugation. And this pellet has been characterized by a nanotracking analysis, by protein dosage, and by functional assays and proteomics. And the uptake was characterized by confocal microscopy and by cytometry. So the first experiment was to compare the production of Giardia intestinale alone, CACO2 cells alone, and the production of EVs during the contact between the hot cells and Giardia intestinalis. As you can see that the Giardia intestinalis produce a large, a higher amount of a higher amount of EBs and host cells. The interaction EBs are producing less. So when we look for the protein concentration, they have similar protein concentration, looking and demonstrating that these EBs has different at the content, have different cargo. And we asked about if this uptake process is a temperature dependent process. And we have seen the same experiment purify the different EVs stained with the PKH26 fluorescent stain, putting in contact with the hot cells, CACO2 cells, by say 6 hours at 37 degree at 4 degree. As you can see that only after this time, the cell was collected and analyzed by cytometry. And you can see that only at 37 degree, we have obtained uptake of the EVs. At 4 degree, didn't uptake any EVs. So to know about if this process is time dependent, we did the same experiment, putting extracellular vesicle from every kind of EVs through the time in contact with CACO2 cells. And we collected in different points the cells, and we have analyzed by cytometry, and you can see similar pattern of internalization of EVs. But if you look carefully, you can see that interaction EVs has been internalized in a quick manner at the first 6 hours. So the next step was to know if this uptake process is a dose-dependent process. So we use 7, 14, and 28 microgram of EVs, every EVs, and we have done to the CACO2 cells and analyzed the uptake by cytometry. And you can see similar patterns of uptake with the different EVs. So this experiment is showing the difference at the first hours of the internalization of the different EVs by confocal microscopy. So you can see hot cells, and at the green, you can see the EVs internalized for the cells that Giardia EVs has less and host EVs has less than interaction EVs. So we started to know which pathway had been used for the cell to internalize the EVs. And for this, we have done experiment with different inhibitor of endocytosis. So MVCD, that is a RAF inhibitor, warmanin, that is a macropinocytosis inhibitor, and cytokalasin D, that is a endocytosis inhibitor, didn't produce any inhibition of the uptake. And dinosaur, that is a dynamine dependent inhibitor, is showing that only clathrin and caveolin pathway has been used for the cell to produce to internalize the EVs. So we decided to know if this effect of dinosaur has been different between the different EVs, and you can see that host EVs, interactional EVs has inhibit in 100% with 100 macromolar of dinosaur. But with Giardia intestinalis EBs, you can use 200 micromolar to produce a complete inhibition of the uptake of these EBs. So we have completed the idea about the different pathway of endocytosis using other inhibitors. So dinosaur again inhibit Giardia interaction EBs in a 100%. Pit-top only, that is a clathrin inhibitor, only produce an inhibition of Giardia EBs, and APA, that is a macropinocytosis inhibitor, only induce an inhibition of interaction EBs. Simvastatin and genistein, that are caveolin inhibitor, has a differential inhibition from interaction or Giardia EBs. This is showing that these EBs are different and have different pathways of internalization into the CACO2 cells, and this summarizes that probably the EBs has been internalized by clathrin, clathrin, caveolin, endocytosis, and macropinocytosis. So this difference of the internalization has shown that probably the CBs has a different component at the lipid profile. So we have decided to do an experiment between Giardia intestinalis and CACO2 cells for in contact for 1 hour and 24 hours. And after, during this time, we collected the EBs produced in this time, and we analyzed the lipid profile by different chromatographies. And you can see that large EBs and 1 hour and 24 hours, we have a higher mobilization of phospholipid in interaction EBs more than hot cells EBs. So the presence of the parasite produce a higher interaction of lipid to produce the EBs. This in 1 hour, phosphatidylcholine, phosphatidylglycerol, and phosphatidylserine are more present in 1 hour at the interaction EBs, and in 24 hours the same. And when we analyze the small EBs produce the same effect with phosphatidylethanolamine, phosphatidylcholine, and phosphatidylglycerol, and phosphatidylserine. But curiously, at 24 hours, at small EBs, the mobilization from interaction EBs has not higher than hot cells, when maybe they have a control of the cell to produce small EBs. And we have done a proteomic analysis of these kind of EBs, and we have seen the different classical marker, clathrin, tropomyosin, tubulin, elongation factor, and at 1 hour and 24 hours, they have some common component like actin, complement 33, histone, GTPase, and alpha-2 macroglobulin. So after this information about uptake, we decided to accept the invitation of Antonio Marcilla, that is a researcher from University of Valencia, Spain, to my PhD student Bruna Sabaque to stay for 2 months in his lab in Spain, and to look for 3D model of CACO2 cells. This 3D model is a culture of 21 days that produce epithelium intestinal cells. And this epithelium, the integrity of the epithelium is measured by a transepithelial resistance using electrode in a transwell chamber. And in this time, we have done a comparison between the release of EBs between the 2D culture cells and the 3D epithelial cells. And in this time, at 21 days, we have done an experiment with the synergies between the parasite and EBs in contact with the epithelial cells to see if we have a breakdown of the barrier integrity. In this time, you can analyze the paracellular permeability, the transport of the different substance, cell apoptosis, the expression of some adenosine that are very important to formate the epithelium at the tight junction level. And in this time, the idea is to use inhibitor of EBs internalization to see if the barrier integrity is kept for the 3D model CACO2 cells. And this is an experiment analyzing the production of EBs in 21 days. And for our surprise, the epithelial cells are producing more small EBs than large EBs. And this is different result that the 2D culture cells that produce always a higher concentration of large EBs than small EBs. And when we have analyzed the breakdown of the barrier integrity using parasite and EBs from Giardia intestinalis, we only see a breakdown of the barrier integrity when we use the parasite with the Giardia intestinalis EBs at 100 microgram and 200 microgram. And the effect of the parasite alone or the EB alone didn't produce a higher breakdown of the barrier integrity. And we look for the transport of the transfluorescein molecule at the transwell chamber, and we always see a higher transport when we use Giardia intestinale and EVs from Giardia intestinale together. So the breakdown of epithelium produces an increased transport when we have a synergies between the parasite and the extracellular vesicles from Giardia intestinalis. This is the other promising experiment when we use the SO1 expression. We look for the SO1 expression that is a very important adhesive molecule that form the epithelial cells at the 3D culture, and we have seen a decrease of the expression of SO1 when we use only the Giardia intestinalis, but the EBs from Giardia intestinalis at 80 microgram at 160 microgram. So this is showing that the epithelial breakdown produces a decrease in adenosine that form the tight junction. And just to finish, the conclusion of this part of the work, at the EBs uptake is a time and dose dependent process. Giardia intestinal EBs, host EBs, and interaction EBs are different and have different kinetics of internalization. Experiment with endocytosis inhibitor suggests caveolin clathrin macropinocytosis uptake pathway in intestinal cells. 3D culture of CACO2 cells represent an intestinal epithelial model in vitro. Release of small EBs is more significant in 3D intestinal cells. A combination of parasite and EBs could be able to break down the intestinal barrier in 3D culture with alteration in paracellular transport and adenosine expression. So the next step is just repeat the experiment with the different uptake inhibitor. And I'd like to finish my talk, and I'd like to thank CAPE, CNPq, UPR, UFPR, Carlos Chagas Institute, my group, and the Antonio Marcillo group that hosts Bruna Sabaque in Spain, in Valencia for 2 months to do the experiment with 3D culture. Thank you very much for your attention, and I will be waiting for questions by email on in other times. Thank you very much.